Hi everyone, I am Sahar Youssef, and today we will talk about proteasomes as a cancer therapy. But before that, what is the cancer? Cancer is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body. There are many signs and symptoms that indicate the disease, like abnormal bleeding, prolonged cough, unexplained weight loss, and change in bowel movement. There are over 100 types of cancer that affect humans. The most common types of cancer in males are lung cancer, prostate cancer, and stomach cancer. In females, the most common types are breast cancer, lung cancer, and cervical cancer. Cancer is the second leading of death globally. In 2018, it caused about 9.6 million, and in 2019, more than 15% of deaths caused by cancer. When we talk about cancer treatment, we talk about many options. Some of them common and traditional like surgery, chemotherapy, and radiation therapy. And few of them new like combination therapy, which is combination of chemotherapeutic agent with miRNA as a cancer treatment. And proteasomes inhibitions. What is the proteasomes? And how we can use it as a cancer treatment? That's what we discover with my colleagues. Thank you.
Hello, I'm Sibal Omari, and I will talk about material and method. First of all, I will start with proteasome. Proteasome is a parallel complex which consists of 4 rings, 2 identical outer rings being formed by 7 alpha subunits and 2 identical inner rings formed by 7 beta subunits as we see. The elementary function of a proteasome is degradation of damaged protein, and it's able to degrade protein in highly regulated fashion because of the process of controlling which protein enter to its inner chamber. The proteins targeted for proteasome-mediated degradation are typically attacked by covalent attachment of polyubiquitin chain, at least for ubiquitin moieties, as we see in the figure, and this process called ubiquitination. It takes place before the protein is being recognized and degraded by the proteasome complex. The ubiquitination is carried out by concerted action of 3 distinct enzymes: E1 ubiquitin activation, E2 ubiquitin conjugation, and E3 ubiquitin ligation. Subsequently, ubiquitinated proteins are recognized, unfolded, and deubiquitinated by the lead of 26S proteasome, 19S regulatory particles composed of ATPase and non-ATPase subunits. The proteolysis takes place at the inner chamber inside 20S core, generating short peptide fragments of typically 2 to 24 amino acid residues. The 20S core consists of 2 outer alpha rings and 2 inner beta rings, each containing 7 distinct subunits as we see in the figure. Each beta ring harbors 3 catalytic beta subunits: beta 1, beta 2, and beta 5. They display different substrate preferences, and their activity are commonly referred to as caspase-like, trypsin-like, and chymotrypsin-like activities respectively. Among the 3 catalytic beta subunits, beta 5 subunit is the major target of current proteasome inhibitor drug via their interaction with the catalytic threonine residue. So as conclusion, we want to use a proteasome inhibitor drug that will interact with threonine in beta 5 in 26S proteasome to inhibit the damaged cancer proteasome from degrading the benefit and damaged proteins by the process I have already explained.
Hello, my name is Lian Abdulsamad and I will talk about the result. The result of the paper was based on some type of proteasome inhibitor. The first one of them is proteasomib, also known as PTZ. This study contains several stages. The first one is early phase clinical trial. This phase had investigated PTZ for its safety and tolerability in over 200 patients with cancer by 2001. It was relatively well tolerated with adverse events like peripheral neuropathy. Despite the remarkable clinical success achieved by PTZ, several limitations have emerged, like many other cancer therapies. A subset of patients respond to PTZ therapy while others do not. Even those who initially respond to PTZ therapy almost inevitably develop resistance over time. The clinical response was typically about 12 months. The second one is carfilzomib, known also as CFZ. It's the second-in-class proteasome inhibitor drug and received its fast-track US FDA approval in 2012 based on its efficiency and safety results in patients with replaced and refractory multiple myeloma. CFZ offers a treatment option with greatly reduced risk of peripheral neuropathy, and also there is some adverse effect including cardiovascular complication, hypertension and heart failure. But overall, these adverse effects are reversible and manageable with careful monitoring. The third one is ixazomib, also known as IXZ. It's the first oral proteasome inhibitor drug. In 2015, IXZ received its US FDA approval as the first orally bioavailable proteasome inhibitor drug, with one administered per week. The fourth one is oprozomib, also known as OPZ. It's a structural hemoglobin of CFZ, and it's currently being investigated in several clinical trials. It showed proteasome inhibitory potency similar to CFZ, and it was effective in decreasing the viability of multiple myeloma cells. The last one is marizomib, also known as MRZ. Marizomib more effectively induced apoptosis in tumor cells from patients with multiple myeloma. In phase 1 clinical trial, the overall response rate of marizomib as a single agent was merely 11%, while the response rate substantially improved to 53% when combined with pomalidomide and low dose of dexamethasone in patients with refractory or replaced multiple myeloma. Without significantly increasing the incidence of adverse event, marizomib has been associated with CNS adverse event. And here is a clinical trial of marizomib on 68 patients, ages between 38 and 82. And this table displays the demographic characteristic. Schedule A with 32 patients, dosing was once weekly on 4-week cycle. Schedule B with 36 patients, dosing was twice weekly of 3-week cycle.
Hello, my name is Dala Abdelhadi and I'm going to start with a discussion. First, let's discuss the drug limitations. The first problem is the different responses of patients in proteasome therapy, where a subset of patients respond to the therapy while others do not, even though those who initially respond to the proteasome therapy develop resistance over time to the treatment. Carfilzomib treatment is associated with different types of adverse effects, including cardiovascular complications, hypertension, and heart failure. But overall, these adverse effects are reversible and manageable with careful monitoring. CFZ remains practically insoluble, so to prepare an injectable solution, the current formulation requires the use of 50-fold excess of beta-cyclodextrin derivative, which is a bit complicated and hard. The last problem is the effectiveness of the drug is very minimal in people with solid cancer, and it needs more study and development to overcome this issue. To overcome these limitations, various potential strategies have been made to overcome the developed resistance to the drug, including the development of proteasome inhibitor drugs based on novel structural scaffold, which gave us a promising results in effectively suppressing the tumor growth in case of breast cancer and prostate cancer. Proteasome inhibitors development has greatly improved the survival and the quality of life for patients with multiple myeloma. Bortezomib is commonly used as a first-line agent in combination with other anti-myeloma agents, which leads to the development of additional generations of proteasome inhibitors. In conclusion, proteasome inhibition has been firmly established as an effective treatment strategy for hematological malignancies and for multiple myeloma. Also, it would be necessary to develop proteasome inhibitor drugs that can afford sufficient access to the proteasomes in solid cancer and to develop targeted drug delivery systems. Further investigations are ongoing to develop therapeutic agent targeting non-proteasomal component of the ubiquitin proteasome system, on their own or in combination with proteasome inhibition drugs. Marizomib drug is more effective in induced apoptosis than bortezomib, while displaying a lower toxicity to normal cells in multiple myeloma and chronic lymphocytic leukemia.